Good evening, viewer. Welcome to our FTV English version. Amazing story. Butterflies. Yeah, many butterflies develop wing patterns that mimic other species to protect themselves from predators. While growing complex body parts like wings involves many genes, the difference between two versions of the same thing, like wings in different colors, is often controlled by variation in one genetic location. A new study published in PNAS by scientists at the University of Chicago describes the inner workings of one such supergene called double sex that helps a species of swallow tull butterfly, that is the scientific names Papilio alphenor, mimic the wing patterns of others distantly related species that are toxic to predators. Using modern genomic sequencing techniques and experimental tools like CRISPR to study the evolution and function of double sex, researchers showed how the supergene gained its ability to control wing patterns by becoming linked with other genetic elements that regulate its own expression. Males and females of these butterflies can have totally different color patterns with pretty much the same genome, but somehow one piece of DNA encodes those different phenotypes, said Nicholas Van Kuren, PhD, a research scientist in the Department of Ecology and Evolution at U Chicago and lead author of the study. What's great about this study is that we identified not only the differences between the two versions of the gene, but also how those differences affected how the gene functions and turns these wing patterns on or off. A supergene is usually a group of neighboring genes on a chromosome that are inherited together because they function to control complex traits such as color patterns and mating behaviors. They are often made up of tens of thousands of individual genes linked together. In swallowtail butterflies, however, the double sex supergene comprises just one gene. Only females in this species develop alternate wing patterns, adding orange spots to their array of white patches to mimic other species. Males keep their standard white patches on a black background. The female limited polymorphism in Papilio alphanor is a classic example of a supergene, said Marcus Cronfrost, PhD professor of ecology and evolution and senior author of the new study. That's why you got interested in studying this, so we can figure out what is responsible molecularly for creating a supergene. Historically, the problem of how they evolve has been kind of intractable, but now we have the tools to dissect them. In this exclusive report, FTV English Delhi, Bharat.